Welcome. In this video, we will compare two important types of microscopes, the compound light microscope and the electron microscope. We will learn how they work, what they can show, and why electron microscopes can reveal much finer detail. A microscope is an instrument that helps us observe objects that are too small to see clearly with the unaided eye. Microscopes are widely used in biology, medicine, research, and many other scientific fields. First, let's look at the compound light microscope. A compound microscope uses visible light and a system of glass lenses to magnify a specimen. The light passes through, or is reflected from, the specimen, and the lenses form a magnified image. The compound microscope has several important parts. These include the eyepiece, objective lenses, stage, light source, and focusing controls. Different objective lenses provide different magnification levels. The total magnification is found by multiplying the eyepiece magnification by the objective magnification. Now let's move to the electron microscope. Instead of visible light, an electron microscope uses a beam of electrons. Electromagnetic lenses are used to control and focus the electrons. Because electrons have much shorter wavelengths than visible light, electron microscopes can achieve much higher resolution. Electron microscopes usually operate under vacuum conditions. Specimens also require special preparation. This means electron microscopy is very different from ordinary light microscopy. There are two major types of electron microscopes, SEM and TEM. SEM stands for Scanning Electron Microscope. In SEM, a focused electron beam scans across the surface of a specimen. Detectors collect signals from the surface and produce an image with detailed surface information. SEM images are often shown with a three-dimensional appearance. TEM stands for Transmission Electron Microscope. In TEM, electrons pass through an extremely thin specimen. The transmitted electrons create an image that can reveal very fine internal structures. TEM is especially useful for studying the ultrastructure inside cells and other microscopic materials. Now we should understand the difference between magnification and resolution. Magnification means making an image appear larger. Resolution is the ability to distinguish two objects that are very close together as separate details. High magnification is not useful if the resolution is poor. A compound microscope is useful for many biological observations. It can be relatively simple to operate, and with suitable techniques it can be used to observe living cells and microorganisms. It is also much more accessible for classrooms and routine laboratory work. Electron microscopes have a major advantage in resolution and can reveal structures far smaller than those normally visible with a compound light microscope. However, they are expensive, require specialized equipment, and specimens generally cannot be observed alive in the microscope. So, here is the quick comparison. A compound microscope uses visible light and optical glass lenses. An electron microscope uses electrons and electromagnetic lenses. Compound microscopes generally have lower resolution, while electron microscopes provide much higher resolution. Remember the two electron microscope types. SEM is mainly used to study surface structure. TEM is mainly used to study internal structure. Let's recap. A compound microscope uses light and glass lenses and is useful for observing many biological specimens, including living material. An electron microscope uses an electron beam and electromagnetic lenses, operates under specialized conditions, and provides much greater detail. SEM gives detailed surface information, while TEM reveals extremely fine internal structures. Understanding these microscopes helps us understand how scientists study cells, tissues, microorganisms, and materials at different scales. Thanks for watching, and keep exploring the microscopic world.